Thank you so much, Joel, for this welcome and introduction, and thank you to everyone here at UW Medicine for having me today. It's been a really fantastic day getting to meet so many of you and get to know this department. You know, I have been overseeing diversity, equity, inclusion, and belonging efforts at NYSCF, as we call it for short, for about 3 years now, and I'm really excited to talk to you about some of those efforts today. And how we kind of prioritize our efforts across the very broad challenges before us. And to start with, I want to tell you a little bit about who we are as an institute. Just confirm I have nothing to disclose as we get into it. We can't really talk about NYSCF without talking about our founding CEO, Susan Solomon, who we unfortunately lost last year to ovarian cancer, but whose legacy is very much still alive. Susan came at stem cell research as a patient advocate, as a lawyer, as an entrepreneur, who realized that there was enormous promise for treating type 1 diabetes, which her son developed through cell replacement therapies. And she decided to apply that incredible acumen to stem cell research. And, she was incredibly prolific at identifying some of the major obstacles in our field and helping to remove the obstacles. So it's not really as much about how much she raised as what she did with it. She didn't, kind of build a building and slap a name on it. She, found ways to invest into supporting the careers of young scientists who were leaving the field as it was under siege at the time. We were founded in 2005. And she focused on ways to develop technologies that would really speed progress throughout the field. And, as someone who recognized obstacles, she realized how much the lack of equity in the field was holding us back and said, we just absolutely have to do something about this. And so this is, her commitment to that is really embedded throughout the organization, and I'll talk about how we embed it into all of our activities as we go. Any of you who wish to learn more about Susan can do so at the link right there. So what we do as an institute, we have a fully functioning research institute, about 75 to 80 scientists and engineers. We have PI-led teams focused on disease research. We have a great technology infrastructure, which I'll talk a little bit about, as well as a repository of diverse induced pluripotent stem cell lines. I should start by saying we're a nonprofit institute. We're independent. We're not under any academic institute. This is part of Susan's desire that we not be encumbered by bureaucracy, but could really serve a role as an accelerator in the field and kind of work with everyone, which we do. And so, two of the other major activities that we have are building a community, which we have done through our grant-making programs for early career scientists throughout the field. And we built a community of over 200 of these amazing scientists. And we also have a lot of education and outreach efforts, including an annual conference on translational stem cell research, which I'd like to invite everyone here to join next year. And we do a lot of other education events that I'll touch on, as they are quite closely linked to our DEI efforts. And so, as we think about how to move all of these more forward on all of these fronts and accelerating biomedical research, it is really just imperative that we account for the impact that systemic discrimination has on all of these fronts that we're trying to move forward. And when I talk about systemic discrimination, I mean, against any kind of group, whether we're talking on the basis of race or gender or ability or what have you. And this plays out in two major ways that I'll talk about today. One is in the scientific workforce, so those of us who are doing the research, those of us who are in the labs and in the clinics, as well as in the patient communities that we're trying to serve. We do see massive health disparities play out, kind of however you measure major health outcomes. And so what we're talking about here, the word underrepresentation is often used. I don't really like to use that word. I think what we're talking about is really a historical exclusion, a systemic marginalization, minoritization of communities that have not been equally participating or have equal opportunities to both succeed in the scientific workforce or benefit from the treatments and cures being developed by many of you in this audience today. This is not going to be pessimistic talk. I think we're in a really good phase in that we have a lot more momentum now than I think that we ever had. And so we have a great opportunity to start to rectify this. So as we level set and think about these problems here, I wanted to share an analogy that really helped me and a lot of my colleagues reckon with what it is that's before us. And so if we consider the analogy of being in a restaurant after the restaurant has closed, you see something of a sort of dual reality. Those who are inside still enjoying their meal see an open sign, and they're enjoying themselves. And whereas those who are outside don't have access to this opportunity to sit down and eat. And they can see very well that they don't have access to that opportunity. Whereas those of us who are inside, we think everything's fine and everything's equitable. And so a good way to think about inequity is that it structures these open and closed signs in our society. Whereas those of us who are benefiting from the open sign don't necessarily realize who isn't benefiting from that. So that is why we cannot recognize those systems of inequity that privilege us. Whereas if we're the ones who are not being benefited, then we can see those right away. I was having dinner with a friend recently who is, when we were talking about sexism in America, and she said, and I should say that she's white, and she said, I really think that sexism is worse in America than racism. And I'm not here to debate this with any of you, but I think this is a great illustration of exactly that for her, she can see the sexism very clearly as a woman, but she can't see the racism so clearly because it's not oppressing her. And all of us have these kinds of privileges and disadvantages depending on our identity. And so we can think of racism as sort of a system of structuring opportunity and assigning value to people based on the social interpretation of how we look, race, which I don't need to tell this audience is not a scientific concept, but is nonetheless really relevant here. But even beyond racism, every structure of inequity unfairly advantages or disadvantages certain individuals, based on identities. And so if we really want this field to move faster, if we really want to build a meritocracy that we think we have, I think we quite don't have that. How can we remove these disadvantages? And how can we really create opportunities for people to succeed based on their merit? So we think of this as a mission critical objective, that building a workforce that is diverse in terms of representation, that is inclusive, in that the individuals in that workforce feel valued and supported and respected, that is equitable in that people have access to opportunities that accounts for their differential starting points, and that people feel that sense of belonging, is going to be a productive, collaborative, innovative workforce. I put this sort of facetious citation at the bottom here because this is backed up by a lot of literature that I'm not going to waste your time going through today because I really want to focus on what we can do about this rather than that, but I'm happy to share that with anyone who's interested. And so, what I'm going to talk about now is the different ways that we inject that perspective, that lens, into the work that we do at NYSCF in hopes of imparting some lessons, some actions, some take-homes that I hope will be beneficial. And I'm happy to obviously discuss any of this in the question period. So I'm going to start with talking about our research. As I mentioned, we really kind of envisioned as an accelerator that capitalizes on the power of stem cell research for modeling human disease and discovering better therapeutics that actually work. And a lot of this builds on Shinya Yamanaka's Nobel-winning discovery in 2006 that you could actually reprogram adult somatic cells from skin or blood into induced pluripotent stem cells that have the ability to become any cell type in the body, or virtually any cell type in the body. And so we have basically taken that and tried to scale it up as much as we can to achieve three of the major areas of potential in biomedical research that stem cells offer. One is disease research, so just really understanding how these diseases play out at the cellular level across a variety of patients, since we can have patient-specific cells that retain patient genetics. One is drug discovery, being able to do in vitro screens or tests, assays for different drugs that are to help us understand what's likely to work for who. And the third, which, as I mentioned, is the genesis for our institution, is cell replacement therapy. So to create healthy cells that are capable of replacing the cells that are damaged or lost in different diseases. And so the way that we've done that is I lied, I do have a video, and I apologize, it's not playing right now. That is my fault. But anyway, there are some really cool robots that are supposed to be showing right now, where we basically roboticized and automated all of these technologies that I have listed here. So that's, the reprogramming of blood or skin cells all the way till they become induced pluripotent stem cells, differentiating those into disease-relevant cell types, genome editing, et cetera. And so, we've developed technology that's allowed us to do that at scale, and that's yielded a pretty vast repository of stem cell lines that we have available as a community resource for anybody who's interested in accessing those that really represent a broad range of diseases. And, the goal from that is to really be able to go from a diverse patient cohort to the stem cell bank to disease-relevant cell types to understand what's going on in all of these different diseases and really get a representative picture of disease rather than just studying a single patient at a time, as most stem cell research studies do, where they kind of focus on a handful, because without this technology, it's pretty difficult to do that. We've also optimized a lot of these differentiation protocols. Most of these are automated, as indicated by the stars here, or automation is in progress. And this is, really allowing us a kind of an unprecedented window into these cell types and how they are affected in different diseases and different genetic backgrounds. We've gone a step further and incorporated artificial intelligence into our automated pipelines to be able to discover entirely new phenotypes in these cells in an unbiased way, using just the AI to guide us on cell-painted images to see if there are, in fact, new phenotypes. We were able to discover new phenotypes of Parkinson's disease and skin fibroblasts, which wasn't necessarily a guarantee, as that's not believed to be an affected cell type. But the kind of scale and the reproducibility that we've spent a lot of years engineering helped us to actually detect some signal from that noise, and we're now trying to leverage that to discover drugs that can reverse any signatures of disease that are found. And like I said, the idea is doing this across a large population. So the Parkinson's study was in a cohort of nearly 100 patients and matched controls, and we're continuing that with other indications. And so through this, we collaborate with a whole lot of organizations. As I mentioned, that was kind of the intention behind building this technology. And we're very, had some great discussions today. Very happy to explore collaborations with any of you. We have these disease-focused research teams, with PI-led teams, somewhat like your traditional academic labs, a little bit smaller. Everybody all sits together. Everybody shares the equipment. It's a little bit of a different culture, but very much, similar goals in terms of just advancing knowledge into how these diseases work. And as I said, our mission is focused on the major diseases of our time. So it is imperative that when we're talking about the major diseases of our time, that we understand that health disparities persist for all of these diseases, and they're actually getting worse. Even though the mortality rates for so many of these major diseases are getting better thanks to medical breakthroughs, the disparity between health outcomes is getting worse over the years. And two examples are shown here for female breast cancer and stroke. There's countless statistics like this demonstrating that patients of color are more likely to die from different diseases than white patients, and the list kind of goes on and on. This also extends to treatment efficacy. Certain treatments don't work as well in populations that have been excluded from research, sort of unsurprisingly. We're talking about really deadly consequences of not including these populations in our research. And so why do these disparities exist? That could fill a day. I'm not going to be able to do justice to that entire topic today, but what I'll touch on very briefly are, that everything at the root of it is really about systemic discrimination, and that plays out in so many different ways in terms of access to healthcare, in terms of socioeconomics, in terms of incredibly justified mistrust that these communities have of healthcare providers of biomedical research writ large. You don't have to read too far back in history to understand exactly why. But the area that we're focused on, and a lot of what I'll talk about is really just focusing efforts. There's a lot of these big issues, but where we think we can make the impact is addressing the complete underrepresentation or minoritization of these communities throughout the research pipeline and throughout the clinical trial pipeline. And so what happens because of that is that our baseline understanding of biology, of health, of disease is incredibly skewed. And that means it's going to be that much more challenging to understand disease, to define disease mechanisms, and discover treatments that will actually help all of the patients that need them. I'm sure this is also no surprise that, if you look at genome-wide association data, that, the vast majority of the samples that have been used are white European. This is also an example from stem cell biobanks for autism research. If it's difficult to read, the blue is white European, unsurprisingly. And so we're talking about, 70, 80% of samples of participants in these studies, being really skewed in this direction. And so one of the things that we're really motivated to do, I described that we have a repository of thousands of lines. Most of them, just like these other ones, are built from white European participants, and that is due to all of the same reasons that we've been discussing. But we're really, motivated to expand the diversity of our biobank. We're partnering with a lot of different organizations who are engaged with these communities and who can, really bring them in as participants and partners rather than, I think we talk about, doing research with them rather than to them. And that's a frame shift that I think we all need to consider as we think about being more inclusive in our research. And so this is something that we're expanding in partnerships. We're always happy to work with more people, and that we hope to create as a very broad community resource, a very well annotated community resource that allows everybody else who maybe is not situated in New York City with a diverse population who does not have access to these robotics, to be able to do research that is inclusive and towards inclusive precision medicine. So I'm going to switch gears a bit right now and talk about the scientific workforce. We've talked about health equity and health disparities. And so for the rest of my talk, I'm going to focus on our work in events and grant making and in our internal culture. So, to start with, when we think about the workforce, some of the major challenges that we're thinking about, and then I'll talk about some of the strategies that we're using. It's by no means comprehensive. And I'll point out that we are an institute of only 120 people. So, we are doing what we can. But this is how we think about it. So implicit bias obviously is one of the key challenges that's at the root of all of this. It leads to systemic discrimination. It's something that all of us have. But it's, one of the ways that, we can deal with it, and that I'm sure a lot of you have gone through, our training and self-education. I think it's important not to get into the self-flagellation on this. Some of these tests can really lead to that and to make you feel, like you're a terrible person for having these biases. We all have these biases. The point is not to feel bad that you have them. The point is to be aware and to use that to inform how you act and how you treat others. So, those are the kinds of things we think about when it comes to training and self-education. We've also, integrated that into our grant review process, which I'll talk about a little later on. Obviously, inequitable access to science education is an issue in terms of seeding the pipeline and getting diverse talent into the workforce. So we have a lot of dedicated outreach efforts to reach populations and students that may not otherwise have access. We all know, especially at an academic institution, that there is deep-set systemic bias when it comes to recruitment decisions, when it comes to funding decisions, when it comes to promotion decisions, tenure, et cetera. And so I'm going to share some of the things that we've done with our grant making in terms of revamping the policies and the processes that we use to understand, to evaluate merit and to evaluate the promise of a candidate that we've found to be really helpful. there is a leaky pipeline. I think this was debated a little while ago. I hope that debate is over, but I'll show you some beautiful, horrible data that will convince you otherwise. We've been making use of our grant-making programs to do some benchmarking data on the state of gender equity across the biomedical research enterprise, which I'll share with you. Toxic environments are something that, I know you know well in academia. We know well in, basically every field suffers from this. And, when I talk about toxic environments, I mean environments that make it difficult, especially for minoritized scientists to succeed. And that's a big problem. It's an entrenched problem. There's a lot of things that need to be done to take it on. We think about it in terms of culture work, changing the culture as much as we can to make it as inclusive as we can. We have an affinity group program I'll say a few words about. We also do a lot of work to build communities that are inclusive, that value DEIB in the way that they do science. And we also advance that through our grant making, which I'll talk about. I have to say as well, I think, when a lot of us take this on, and even just conversations I've had with some of you advocates here today, the enormity of the task can be very daunting. We're talking about problems that are way bigger and way older than, any of us are and that any of us can remember. So, I think one of the biggest challenges is where do you even start? How do you even take this on, right? And so, as I've mentioned, I think being intentional is really, really important. Accepting that there's only so much that we can do is really important, and working with others, learning from each other, who's doing, the great work. And I've learned from so many of you today who are really making an impact. Raising awareness and discourse, I think through meetings like this, and I'm, very grateful for the opportunity to do that here, and collaborating with those who have, kind of learned things the easy way or the hard way, to figure out sort of those actionable strategies. So it's really about that sort of prioritization. And I'm continually heartened by how many people are taking this on in a very intentional and thoughtful way. So, we launched the initiative on women in science and engineering at NYSCF about 10 years ago. This was, the moment I alluded to when Susan said, we've just got to do something about it and fed up with this. Nothing's changing. And so, she set out to assemble a working group of, sort of thought leaders all over the academic pipeline and define actionable low-cost or no-cost strategies that institutions could take to improve gender equality. And so this is what they came up with. It was, is published, this is before my time, this was published in Cell Stem Cell in 2015. And these strategies, we've been, heartened to see that a lot of institutions have been taking this on since we published this data. One that I want to zoom in on a little bit is creating an institutional report card for gender equality, which we took advantage of our grant-making program to do. And so we thought, this is something that we require every applicant to our extramural grants to complete on behalf of their department. So their department, chair, administrators, et cetera, have to gather this data, and I'll go through what that data is. But effectively, the idea is to shine a light on, just what the state of gender equality or inequality is. Hopefully, encourage institutions to combat, this really leaky pipeline, to identify other best practices that institutions are implementing, and as I said, providing a benchmarking data set so that we can really understand what the state of things are, how are things improving, what kinds of interventions really actually work. And so through our grant-making programs, we were able to collect data from over 500 institutions in 38 countries in a 4-year span, which we published in Cell Stem Cell a few years ago. So I promised you some beautiful, horrifying data. The leaky pipeline is very linear, alive, and well as of the collection of this data set. We see above 50% women at the undergraduate level, and across these 500 institutions, there was a very, very clear decline down to the full professor level at about 25%. In a third of the institutions that we received data from, women were only less than 10% of tenured faculty. Unsurprisingly, this also corresponded with the fact that women were poorly represented in decision-making committees and in positions of prominence. And that goes to external seminar speakers, promotion committees, strategy committees, recruitment committees, finance committees, award committees. And unsurprisingly, if you don't have any policies to actually promote diversity or gender equity on these decision-making committees, surprise, they aren't actually going to foster that much gender equity. I'll say, as a disclaimer, this was a few years ago. I'm hopeful that things are changing. We did not see them change in the 4-year span of that data. There was no kind of statistically significant improvement. But we did see the emergence of some of these policies that I mentioned, some of the actionable strategies that I mentioned. So we're going through and analyzing the data now and hopeful that I'll have some better news to report at the end of that. But this is, and this is where we're just talking about gender. Of course, the situation gets much more extreme and exacerbated when you look at things like race and ethnicity. We don't have that on our report card. That is data that we have not figured out a good way to be able to collect. Even collecting this data was not easy, and we had many resistant institutions and departments who did not want to provide the data and just didn't let their faculty apply to our grants. So, and I should say as well, we did not base any grant decisions on this data because everybody would fail. So that was definitely not part of it, but nonetheless. Okay, so I'm going to talk now a little bit about our events and communications efforts, which we have, we have a role as a convener in the community through our annual conference, through all of the events that we hold. And so we like to use this in several ways to advance DEIB. One is to, engage everyone on the issues in DEIB and get that, kind of dialogue and engagement going. Another is that, we have a platform that's seen by a lot of people, a conference that, 500 people attend, webinars that thousands of people attend. We try to make sure that we represent as diverse voices and speakers on our platform as we can. And we also try to broaden the access to science education. We do this through several different education programs that we have, a really great summer internship program, a sort of stem cell boot camp that we do for high school students from particularly underserved communities. And we achieve this by partnering with a lot of organizations that have done great work in identifying the students who need this education the most and making sure that we reach them that way. Over the pandemic, silver lining, I guess if we have to find one, is that we went virtual and we're able to reach a lot more people. So we put together a lot of educational events for students, for teachers, for parents, some career development for the scientific community. We hold lay-friendly expert panels on a variety of topics that we want to have a dialogue with the public on. We also, as I said, spotlight specific DEI issues in our events, make use of them to, really focus on, for example, the issue I mentioned of diversifying stem cell biobanks, reflecting on how far we've come in terms of women's history. We had a webinar on that last year. As I mentioned, we hold an annual conference on translational stem cell research that, we'd like to invite everybody to come to, and we showcase DEIB topics as part of, we have a single track, so this isn't like at lunch in some room that nobody can find. This is as part of the main session that if you're attending the conference, you are going to, and so we've, kind of rotated the topics across the years. Just a couple of weeks ago, we held a discussion about diversifying biobanks and all of the trust that we need to build back with the communities that have been excluded from research. And a lot of these are, I think all of these, except for the last one, which we're just finalizing, are available on our YouTube channel if you'd like to watch any of those. In terms of internal culture, I'll just go through very quickly, but the idea of our culture work is to make sure that we're engaging all staff in giving them a voice in how we achieve DEI. So, from the leadership down to the most junior staff, that we're offering training opportunities that will actually help to move the needle and to really just build as kind of inclusive and supportive of an environment as we possibly can. The main thing I want to talk about here is our affinity groups. This is something that I believe you also have here in the departments at UW. And so we've set these up to, just really build community and, allow networking, resource sharing, et cetera. Each of these groups supports a particular marginalized identity in STEM. So we currently have groups representing gender, sexual, and ethnic minorities. We include allies in all of these discussions. We have a lot of allies who participate. It gives, like I said, everyone an opportunity to have a voice. Here are some examples of activities that we've held recently. We have some regular meetings on topics that are defined by the groups. We have some kind of emergency meetings when something really bad happens that causes a lot of feelings, and we, kind of build a support room for everyone to sort of share their feelings. And we also do kind of, fun things like movie nights and going out for international food and honoring Heritage Months in fun ways because, I think it's really important that the efforts we do for culture building focus on creating an environment of support and celebration for all of the communities that work with us and, integrating into our group rather than stigmatizing by focusing consistently on the horrible things that are happening around racism, et cetera. It's a fine balance, but it's something that we've found has been helpful, and has helped to create that environment of inclusion. We also talk about, I talked about toxic environments. I think I wanted to get very specific about some of the narratives that create a toxic environment that I think we all need to do a little bit of work in dismantling, because the more they kind of take root, the more they can perpetuate these kinds of exclusive environments that make it difficult for everyone to succeed. So I think we've all been told this, that science is a meritocracy, that, if you struggle, then you're just not cut out for it. We're actually not on a level playing field, and I think we all need to acknowledge that. We focus way too much on the data, on the results, on the papers. The well-being of our colleagues is important. That is an important part of culture work. Making sure that, as a mentor, that you're checking on the well-being of your mentees and your other colleagues. That is just as important for advancing the productivity of an institute as checking the data. We also use lazy proxies, like the number of Nature Cell Science papers, the number of papers generally. What's the story behind those papers? That's actually a much more important indicator of whether that person is likely to succeed and what the magnitude of their contribution to science is. And so that takes me into the final phase of what I'll talk about today, which is our grant making, where we think we can really sort of move the needle in how the community is looking at these kinds of criteria. So the goals of our grant-making efforts, our DEIB-focused grant-making efforts, are to make sure that we have applicant and awardee pools that are as diverse as possible in every dimension, not just, ethnically, but scientifically, geographically, et cetera. I mentioned the benchmarking data earlier. We've been working to reduce bias in the review process and to ensure that we are really selecting based on merit. That also does require evolving to an extent our definitions of merit and potential, and I'll talk specifically about what I mean there. And as I mentioned, the goal of our grant making, in addition to supporting these really talented scientists, is to build a community that can work together, that can tear down a lot of these silos that are impeding progress. And so in that community, it's important to us that we are advancing DEIB values, and that community is doing so, in their own labs and institutes. And so some of the strategies we've been using, I went over the report card. Obviously we try to have as diversified review committees as we possibly can. We wrote an implicit bias statement that's read at every meeting and every time we connect with our reviewers. That was something we did many years ago that many other organizations have borrowed from. Our funding model is also flexible funding, so people can use it for things like childcare, for extra hands, for all of that kind of stuff that can really help to, again, level the playing field. But what I'll focus on in the next couple of slides are some more recent elements that we've added to our RFAs that we think have moved further to leveling the playing field. I'll also mention, just here that we eliminated reference letters for the junior PIs, because that has actually been documented as a pretty strong source of bias. You get big shot name, big shot from big shot lineage. That can, that's pretty much the only impact that it has is like, oh, they know so-and-so, and so-and-so wrote a nice letter for them. And that just creates this sort of lineage bias that is not helping you understand the merit of the candidate. And so we, like many other prominent grant organizations, have removed this from our evaluation. It also takes the burden off the applicants and reviewers a little bit, which is just a bonus. So as I mentioned, through our grant-making programs, we've built this network of over 200 scientists all over the world. You can learn more about that at the link. We've had some really prominent investigators who've come through our program and founded really exciting companies and made really exciting breakthroughs. And we're really fortunate to have that. Derek Rossi is one who I'll highlight, who we funded in 2010, shortly before he founded Moderna, and he is now our interim CEO, has been on our board for a couple of years. But we've had a really, really excellent crop, and we're very grateful for them. And a lot of them have now come to serve as reviewers. But so the new grant elements that I mentioned for the RFA, one of the goals is to account for any undue hardship that an applicant may have faced due to their identity. If that's due solely to their identity, that's something that we're not really accounting for based on the way, reading a biosketch or reading a list of publications. And so we've included an optional self-identification statement where applicants can identify as part of any minoritized group. We don't try to slice and dice all of the categories. The definitions of all of these categories are problematic and not particularly informative. And so we're not really interested in that. We're just interested if, there is any kind of marginalization you've experienced due to your identity, you're able to talk about how that affected your careers. And so we instruct our reviewers to use this information as they evaluate track record. So in a sort of, past evaluation, as you look at the biosketch, you look at the publications, you also look at this and say, have they made it through, something really difficult or challenging? That is actually a proof of resilience, of roads traveled, and that actually speaks to the potential of a candidate to succeed, alongside their publications. The other goal is to reward advocacy. Like I said, we want to instill these values in the community that we're building, and so we, like many other institutes, have instilled a mandatory commitment to DEIB statement, where applicants will detail anything that they have done, are doing, or plan to do to advance DEIB in their labs, in their institutes, and community. Of course, this has nothing to do with identity. This is just really about your actions and what you're doing. And so we use this in a very intentional and dedicated way to tier applicants. So we come up with these sort of 3 tiers of advocates who really go out of their way, sympathizers who understand the assignment or are kind of doing what they can do, and non-participants whose radar it's not totally on. And we take that information into account and use it to structure the review process and the order in which candidates are discussed, so that we are advantaging those who are in Tier A. So a couple of pieces of data I'll share in my last few minutes here. One is that we've been happy to see that actually our minoritized applicant pool has been increasing over time in terms of both gender identity and self-identification. We saw quite a big jump one year to the next. I won't say that that's entirely due to actual more diverse candidates coming in, but maybe more people who are willing to check that box after they understand what it's being used for. I think there's a little bit of that going on there that we can't totally rule out. But nonetheless, we're happy to see, kind of any increase, and that people, that more diverse candidates either feel comfortable applying or identifying themselves, when they apply. One of my favorite activities over the last couple of years has been reading the commitment to DEIB statements and seeing just the outstanding things that this community is doing. And like I said, we're funding junior PIs, we're funding postdocs, And nonetheless, the things that they are doing are really, really inspiring. And this is kind of a crude sort of summary of the topics and the kind of areas that they work in. But these are all things like advocating for policies that will promote fair review in your on your tenure committee. Going out and designing a course for underserved students. Going above and beyond with your self-education to really to elevate that, and so on and so forth. What we are also happy to see is that our review process seems to be doing what we intended it to do, which is to elevate minoritized and DEIB committed candidates. So what this graph is showing is sort of our tiers. As I mentioned, A is your advocate, you're sort of above and beyond. C is your non-participant, B is in between. And we see that as we progress through the review process, we are, kind of enriching for those very committed candidates who are, really going above and beyond to advance DEIB in their communities. We also see a similar sort of trend for gender and enriching for female and non-binary candidates, as well as those who have self-identified. And I'll say that this isn't by instruction. We are not telling the reviewers that we have to fund, minoritized candidates, that we have to fund committed candidates. This is really what we think is by emphasizing the importance of this alongside scientific merit as additional factors that we encourage them to take into account. And I have to say that our reviewers wholeheartedly embraced this this approach, and they were from kind of all career stages, all backgrounds themselves. So that was really heartening to see. The last piece of data I'll leave you with, which made me very happy, is that we actually did see a correlation of scientific merit scores with these tiers, with the level of contribution, right? So what does that tell us? One, it tells us that, as I said, our reviewers are taking this in and taking it to heart. Another thing that we found is that no Tier C applicants have received the award, so none of these non-participants. Again, that is not by instruction. We do not say reviewers do not rank these people highly, do not score them highly. That is just kind of what happened. So a few ways to interpret this, but I think one that we can take is that what we're seeing is that the best scientists are those who value diversity, are those who understand that this is how the ecosystem works, that if you create these better environments, if you're committed to these better ways of doing science that are inclusive, that you are going to succeed more as a scientist. And so that's, a heartening correlation to see. I look forward to digging into more data as it comes in over the next couple of review cycles. So I'm going to close with some take-homes and some actions that I want to leave you with. So, the first is that if we really do level the playing field, if we really do make this into more of a meritocracy, everything that we do is going to move faster. I know that all of us are facing the same challenges in our research. We're frustrated by how slow going it is. We should take every tool that we have to accelerate things. The second is that, for DEIB to be really successfully implemented in an organization, it does really need to be top down. It does need to be throughout the organization. It does need to give everybody a voice in sort of how it's done. There's a lot of nuance. There's a lot of dynamics. There's a lot of things that we can't purport to understand from somebody who's at a different career stage from us. And so that, kind of integrative approach is really, really important. At the same time, our own individual contributions are really important. And as we grapple with those, how we can best individually contribute, that self-awareness is really, really important. So, think of the privilege metaphor that I mentioned. Remember that, because we're blind to it. I still forget it every day, even though I absolutely love that metaphor. It's something that, we just have to be aware of. And, being comfortable with the discomfort that it gives you is also something that we just need to get over. As I mentioned, we can't boil the ocean. There's so much to do in this space. It's so important to be able to prioritize, to be intentional, to collaborate, join forces with others doing this good work. And I want to leave with a note of optimism. I think this work is too often framed as being about guilt and being about feeling bad and trying to atone for the sins of the past. I think that by adopting a more constructive action approach, even if those actions feel small, even if they feel incremental, everything is important, everything counts. And by dragging yourself down into the guilt and negativity of it, I think that is where progress gets impeded even more. It needs to be a collaborative approach. It needs to be constructive. And so, remembering that we're at this inflection point now, these conversations didn't exist 5 years ago, as I certainly never heard anything of it. And so this is very positive. We've got sustained momentum, and so we have the opportunity to follow through on it. So I'm going to leave some resources here. I've been assured that they will be circulated to all of you through the department, because I don't expect anyone to write this down. I want to acknowledge the incredible team that is behind all of the work that I've been presenting, and our really outstanding collaborators, funders, and more. And I want to encourage you all to keep in touch, join our community. We'd love to have more people in, collaborate with us, reach out anytime if there's any way that we might be able to help you or work together, whether it's on DEIB or science or technology or anything. So I want to thank you all for your attention. I'm very happy to take questions.
Thank you. That was a wonderful talk. I have a question about all of the various different activities and things that you've done, the metrics that you're keeping and all that. In the light of a recent Supreme Court decision that we're all aware of that sort of overturns affirmative action, and you may or may not be aware that we have a law in Washington State, I-200, that forbids discrimination, consideration, of such, for example, race in hiring or admissions. How are the activities and things that you're doing there, do you think, how do they relate to those rules in place now or things that could happen in the future?
Thank you for that question. I'm just going to repeat in a bit of a paraphrase in case it wasn't clear for those online, was just, in light of all of the recent Supreme Court rulings around affirmative action and the I-200 law here in Washington State, how does that sort of interact with what I presented here about our policies? And so, I'll say, first of all, I think the decision has complicated a lot of this work for a lot of us. It's made it a lot more difficult, even though, I think we all understand that that being more diverse in our communities and being more inclusive is going to be important for the sort of bottom lines of everything that we're trying to achieve. That being said, first of all, what I've been talking about is in our grant-making initiatives. And so that does not, that's not affected by the SCOTUS ruling because it is not about employment and it is not about student recruitment. But, nonetheless, I think that there are kinds of lessons within what we're doing that I hope are translatable. A lot of the things that we've implemented has been in discussions with, academic institutions like yours on how they do recruitment and how they do promotion and all of that. And so I think it is possible to integrate some of these elements without, as I said, we don't instruct our reviewers to say, oh, you have to pick a minoritized scientist for the award. That's not what we want to do at all. What we're talking about is really changing the way that we define merit, to understand those roads traveled, to factor those in, to say that somebody who's committed to DEIB is a contribution to the scientific community, to progress, that should be considered as additive to their track record in publications and awards and so on. And so I think it is possible to implement some of these. Of course, I'm not a lawyer. I'm not going to tell you exactly how I think that should happen. And that's very dependent on context and your own culture. But I'm hopeful that there are some lessons in there that can be applicable across. I hope that makes sense. Thank you. Yes.
So there's a preprint that just came out looking at K99 R00 awardees. Mm-hmm. And they found that there are no K99 awardees in the history of the program that come from historically Black colleges and universities, which is kind of stunning because that's where the majority of Black doctors, scientists, and engineers come from. And so, one interpretation of that might be the resources that are available for environment for that sort of section. And so how do you incorporate that into the granting process? Because, I'm in an institution, right, which has pretty much everything at my fingertips, so it's really easy for me to say I can do X, Y, and Z with the help of, these core facilities. But what do you do for scientists that don't have that at their beck and call?
Yeah, that's an excellent point. I'll paraphrase again quickly. Is a recent preprint showing that there have been no K-99s awarded to scientists from historically Black colleges and universities. That's really disheartening. I had not seen that preprint. And then in terms of the potential access to resources and environments there, that obviously are a boost to people getting those awards. So I think, in terms of our grant making, what I can say is that we don't have that like kind of environment criteria like NIH does as explicitly, which, by the way, I've heard that they are actually phasing out to help to address exactly your point of those kind of inequitable access to these resources. So that's not something that's explicitly considered in our review process. Whether it's implicitly considered is an important one. I think we try to account for it through the measures that I've mentioned. I do have to also say we haven't gotten a ton of applications from less sort of resourced institutions and universities, and it is something to really consider. So thank you for bringing that up.
Let's have a question about the leaky pipeline.
Yes.
So, what you're looking when you show that graph, it's a snapshot of the current status. And so I guess my question, I realize there's attrition along the way. There's no question people drop out along the way. But I guess the question I have is there reason for hope that, this potential bolus down there at the end where people are undergraduates and greater than 50% women, for example, is there any hope that that's going to eventually shift and raise the numbers as time goes on, as people get older, essentially?
Yeah, I'm hopeful that will happen. Certainly, the question was around the leaky pipeline and whether there's hope that the kind of undergrad seed will go throughout. I think what we're seeing in that snapshot of the data is really a result of toxic environments that are not equipped to allow women to succeed in science or other kinds of minorities. And I think that is very clearly why we're seeing that drop off. And that's it is a snapshot, but it's also over a 4-year period. So it's and it, as I said, it didn't get any better. So I think there is something to the age sort of situation that's happening, that a lot of those scientists who have gone through all the way to full professor, when they started out, maybe it wasn't 50-50, but I think it's unquestionable that there are environmental factors in just making it much more difficult for women to succeed. But as I mentioned, a lot of institutions are adopting some of these strategies. I think I'm hopeful that the picture is going to be different when we analyze this new batch of data, though I will also note that it's since the pandemic, that includes the pandemic years. So those have been, well documented to introduce many additional challenges to women succeeding and in the research environment and publication hits and all of that. So I think it comes down to really changing the way that we do these review processes and these tenure committees, these promotion committees, factoring in roads traveled, factoring in other types of adversity when you're reading out that person's list of accomplishments. We've also had some conversations today around how can you factor in the other ways that faculty are contributing, like through mentorship, through what's also often known as shadow mentoring, where you're kind of mentoring someone who's not necessarily in your in your lab, or you're not on one of their committees, but where you're providing that sort of extra help. How can we revise our processes to reward and incentivize that kind of behavior, because that is important for success? So I think it comes down to that. There's another question back.
This is a bit of a follow-up question to that, and maybe a bit of a two-part question, but sometimes having been on some sorts of search committees or other sorts of committees, people will talk about, oh, this person is more diverse than this other person. Like, how do you handle or approach when there is some sort of trying to do some sort of comparison, and sometimes people are, I think, trying to quantify kind of diversity in a sense. And what do you do when that happens? And then separately from that, but also in a related vein, sometimes there can be people who are, and I love the part where you talked about the advocate versus what is it, like supporter,
Sympathizer.
or sympathizer,
non-participant.
in terms of putting those tiers. But sometimes we see people who are great advocates, but maybe are not from an underrepresented minority. And then, so how do you kind of square those two things when you're evaluating the contributions to diversity?
Yeah, great question. So I'll tackle the second one first. So where you talked about how you kind of square away both the kind of contributions to DEI and commitment with someone not being from a minoritized identity. We treat those as very separate things in the review process. Because I think they are very separate things. You can absolutely be an outstanding advocate if you have grown up with every privilege known. Whereas, you can also be, a minoritized person who maybe does not engage in DEI efforts. I should say we factor in the fact that, a lot of minorities are tired of trying to, there's the minority tax. It doesn't mean that just because we're minorities that we need to do all of this work. And so we don't want to unduly penalize anyone for that either, right? So you bring up an important point. The commitment is something that we consider as its sort of own thing. We kind of adjust the scoring of that commitment if somebody has really kind of gone through some very terrible things in their own life, then we don't maybe judge them quite as harshly in terms of how much they need to be contributing as an advocate. So that's sort of one way we account for that. And then, the other way is just, like I said, that these are kind of considered as separate pieces. The self-identification is more about track record and, someone's previous achievements, whereas the commitment is kind of more about the present and future sorts of contributions, if that makes sense. And sorry, your first question, if you could.
It's kind of related to that, like how much weight do you put to each of those? And then like people talking about quantification of diversity.
Right.
But then that's also tied up with intersectionality.
Yes.
In a sense.
Yeah, so quantifying it. I think that's a really important thing that we've thought about a lot too, is like, it's hard to quantify these things, of course, right? It's, you don't want to put a score on how difficult somebody's life has been because they're a minority. That's a hard thing to put a number on, and you're never going to do a good job at it. So we treat it as a bit more of a subjective criteria, which, arguably so is looking at a biosketch and saying like how good of a scientist they are. It's just that when you look at the biosketch, you're also looking at the self-identification, and you're using all of those qualitatively to come up with, your assessment of the candidate. And kind of similarly with the commitment, that's why we kind of go with the tiers rather than really trying to score it. And it's a bit more sort of qualitative in that way.
I have an intersectional stem cell and diversity question.
Cool.
So I was really excited about the efforts that you're making to broaden your patient sample intake. But one of my questions is that iPSCs, you reset the epigenome essentially, and so you will still catch genes of recent ancestry, right? Differences there, but it may be that the environmental factors that are also very much playing into disease outcomes are being erased. And so, do you guys? Given the sort of the great reach that you've been able to achieve, do you have plans to also sort of get into some of the epigenomes prior to sort of that reset, and maybe even to compare them before and after, to see if you can kind of determine, what's being modified that's creating some of this disparity.
What a fascinating scientific question. I think, well, we should write that grant together. That sounds great. For those who may not have heard it, the question is around stem cell reprogramming, kind of resetting epigenetics, and potentially losing some of the environmental factors that might be determinants of disease outcomes or treatment responses. That's absolutely a great point. We don't currently have a way that we're looking at that. A lot of the the samples that come in are fibroblasts, and so they do theoretically capture a little bit more of that environmental influence than what you've sort of wiped out in iPSCs. We have not really done a before and after kind of comparison, but that is exactly the kind of thing that might be cool to do in a collaboration. So definitely open to discussing that. Yeah.
Maybe one more, a little over time. We have another question.
No one final questions, but we do have a large online audience.
Great.
Good, thanks.
Thank you.
Thank you so much.